Welcome to part 2 of week 2, still phase 1. This next video we're going to be just focusing on bullet point number 3, describe the excitation contraction coupling, including neuronal control and sequence of events. This other part of bullet point 2, those major parts of the muscle fiber and their function, we're going to cover that in the next video. Once we get through excitation contraction coupling, I feel like the order of it is a little bit better. So with excitation contraction coupling, specifically what we're talking about is how the muscles get excited, their excitation. So when the action potential comes down from the nerve, goes into the muscle and causes contraction. That excitation and contraction are coupled together. Okay? A little bit of vocab, kind of what we're going to be generally talking about. There wasn't a great place to put this, so I figured I'd start with it. We have something that's called a motor unit, where we have a specific nerve. If we look at any nerve, it comes down, it's going to innervate nerve cells. Remember, nerve cells are also known as muscle cells are also known as myocytes or muscle fibers. So a motor unit is one neuron and all of the muscle fibers or muscle cells that it innervates and controls. Okay? So different muscles have different numbers of motor units, and depending on the action that you're trying to do is going to determine kind of how the motor units are going to be activated. Again, we're going to come back to this idea a little bit later when we talk about the different types of muscle, how big the motor units are, and how they get recruited based on the muscle typing and all of that stuff. That'll be in a future video. This is typically a really good question. It's a very, very simple question. What is a motor unit? It is a specific neuron and all of the muscle fibers that it innervates. Okay? This is a key point as well. We'll get into this later on a little bit more. There's an inverse relationship between the size of the motor unit and the degree of control. So, for example, our fingers, we need really, really fine motor movement, very, very fine control. So the motor units are very, very small. There's a lot of nerves that go in innervating smaller amounts of muscle fibers in our fingers so that we can get those very, very fine movements compared to something like, let's say, the quadriceps, which in reality don't have a lot of jobs other than, that gross overall movement of walking. So those tend to have larger motor units without a ton of very specific control. We're going to start by zooming in to this region right here, what's called the neuromuscular junction. This is where, remember, we talked all about the nerve cell, we talked about the axon, and how you have an axon terminal. These represent the myelin sheath, the Schwann cells wrapping around it. It's going to come all the way down, and there is an axon terminal, which is going to connect to the muscle tissue. And we're going to look at how does the message that is coming down from the neuron pass into the muscle and cause the muscle to contract. We're going to be jumping back and forth to this a little bit. This is kind of all of this written out. Feel free to take a screenshot specifically of this page. This is going to give you a good kind of overall rundown. But we're going to go through all of this slide by slide. So our very, very first step. The action potential travels along a motor nerve all the way to its ending on the muscle fibers, and it's going to activate voltage-gated calcium channels. So there's a nice picture here. So here's the axon of our motor neuron. It's coming down, where it's going to meet with the muscle. This whole thing is called the neuromuscular junction. And as the action potential is brought down, remember we have all the voltage-gated sodium and potassium channels all doing its thing as it goes down. When the voltage change reaches here at the bottom, we have these voltage-gated calcium channels. The voltage-gated calcium channels are going to open when the action potential reaches the terminal, and calcium is going to flood inwards. We have these little packages of a chemical known as acetylcholine. We've heard of acetylcholine a couple of times in biochemistry, as maybe a little bit as well in BCT or DAA. But we have these small vesicles or containers holding the acetylcholine. We're not going to get super deep into the specifics of this, but basically when calcium enters, it's going to come and interact with that vesicle of the acetylcholine and cause it to exocytose, or to leave the axon terminal and enter into the specific word, it enters into the neuromuscular junction, into what is known as, make sure that I get the right word, the synaptic cleft. Yeah, enters into the synaptic cleft. Okay, the acetylcholine, we mentioned before in week 1 that we have those ligand-gated ion channels as well. This is where those now come into play. All of this was all voltage-gated up here. The acetylcholine gets released into the synaptic cleft, and it's going to come and bind on the motor end plate onto these ion channels, and these are our acetylcholine receptor site. These are our acetylcholine ligand-gated cation channels. They call them cation channels. It's a little bit different. I want to make sure that we understand this specifically. They are not super specific to just one ion. Like when we talked about up here in the axon, we had the sodium-gated, the voltage-gated sodium channels and the voltage-gated potassium channels. These ones are just cation channels. These are ligand-gated cation channels because they allow the passage both of sodium and potassium, as well. They will let other things come through as well, other positively charged cations, but mostly the sodium to come in and potassium to leave out. Okay? What this does is this begins to change the overall charge here within this area, and similar to how the message propagated, how it extended in a nerve cell, the same thing's going to happen here in the muscle cell. It's going to travel along until it reaches a point just outside of this neuromuscular junction where there are voltage-gated sodium channels lined up along the edge, lined up along the membrane of the muscle fiber. The name of this membrane is the sarcolemma. Okay, we'll see that in a couple more slides when we look at some more of the anatomy. This plasma membrane of the muscle fiber is the sarcolemma. So the signal is going to come over the voltage-gated sodium channels on the sarcolemma, are going to open, and the action potential is going to continue to propagate in the muscle fiber. So here is just kind of showing that acetylcholine release, the action potential comes down, calcium channels open, that's attached to the vesicles, and we get that release of the acetylcholine, the exocytosis of acetylcholine. Here again, this is just another closer picture, a little bit more detailed, that you can see of all of this happening. We have these little things called junction folds. These aren't super important on all of this, like the nitty-gritty specifics of it. Just look at what your professor says, how deep they go into detail, and look at it compared to that sort of detail. Here are those acetylcholine-gated channels. Like we said, mostly sodium, but also some calcium, and we can also have potassium leaving. So sodium, calcium, potassium. Remember, these are acetylcholine-gated cation channels. Remember, these are not voltage-gated channels. These are ligand or chemical-gated channels. Specifically, what ends this sort of process, because of course we don't want the muscles constantly receiving stimulation over and over and over and over again. Otherwise, we'd have uncontrolled contraction, we'd have continuous excitation and continuous contraction of the muscles. So an enzyme called acetylcholinesterase gets released into that synaptic cleft, breaks off that acetylcholine, breaks it down, and it gets reabsorbed and recycled back into the axon terminal to be used again. Again, here's that whole neuromuscular junction. Now, the next portion, like we said, we have our plasma membrane that is along the muscle fiber called the sarcolemma. Inside, kind of like when we looked at cells, we had cytoplasm. We have sarcoplasm, that inside liquidy kind of inside, called sarcoplasm. And the sarcolemma is our plasma membrane. We get the voltage-gated channels, voltage-gated sodium channels and voltage-gated potassium channels, to very, very similarly have the action potential run down just like it did in the nerve. It's going to follow a very, very similar process here with those voltage-gated channels, so that it propagates and continues the signal all the way across until it reaches a point called a T-tubule. We're going to take a step back a little bit, and look at what is called a little bit more kind of anatomy, general type stuff. With around our muscle fibers, each of these is a myocyte, myofiber. Correction, these are the myofibrils. This whole thing is a myofiber. We have T-tubules, and we have what are called terminal cisternae. The terminal cisternae are back up. The T-tubules are the continuation. They call them invaginations, so like an insertion, a tube channel going in. The T-tubules are a continuation or extension of that sarcolemma, and those are filled with extracellular fluid. They go deep into the muscle fiber, and they branch and form the networks to connect all of the myofibrils, so that way we can get the signal, that action potential, all the way into all of the muscle. On either side of the T-tubule, the transverse tubule, we have these things called sarcoplasmic reticulum. Okay, the sarcoplasmic reticulum, or we'll also call these terminal cisternae. They are running along either side of these T-tubules, and we're going to see what's important about these terminal cisternae in a second. Main function of our terminal cisternae, of our sarcoplasmic reticulum, is going to be calcium storage, when we're going to want to release calcium into the muscle fibrils in order to cause contraction, which again we're going to have to double back a little bit and look more at the total anatomy, like the super specific anatomy of these myofibrils, which is that second bullet point that we mentioned. There's a couple more things that we need to mention. That's what specifically is happening in this area when our signal goes across the sarcolemma and goes down into the T-tubule. That pairing of the two terminal cisternae and one T-tubule is known as a triad. Okay, so if you ever see that word triad, that's what it's referring to, the two terminal cisternae and the T-tubule. There's some pictures of it. So here's the T-tubule, the terminal cisternae of the sarcoplasmic reticulum, and we have these receptors that are voltage receptors called dihydropyridine receptor. Okay, we'll also call these DHP receptors, and these are voltage sensors that are along the T-tubule that have then some connections to ryanodine calcium channels. Okay, So we have DHP being the voltage sensor, and then ryanodine calcium release channel that is attached to the terminal cisternae, and both of these are attached to each other. So the DHP receptor is a voltage sensor. This ryanodine is known as a mechanical channel. So there's going to be this nice little animation. So we have our action potential. We have a calcium pump that's responsible for bringing calcium back into the sarcoplasmic reticulum. When that calcium, that once it is inside of the sarcoplasmic reticulum, is held by a protein called calsequestrin. So our action potential comes down the T tubule. The voltage changes. Our DHP receptor senses that, opens our ryanodine calcium channel, and calcium comes out and is released into that general area, and later the calcium pump brings it back in. This right here is going to be representing, this shape is going to be representing the inner workings of our muscle cells within something called a sarcomere, and that is actually where we're going to end on this video and take a step back and now look at some of the anatomy of those muscle fibrils, finishing bullet point 2, to then see why that release of calcium then triggers contraction.